Hello students. So you all know that our chapter molecular basis of inheritance is going on, and we'll study today gene expression in prokaryotes. And a model is there, Lacopron concept, we'll study about it. So what is gene expression in prokaryotes? So there are a number of studies on metabolism of bacterium Escherichia coli. Two French scientists, Jacob and Monod, biochemist, in 1961 proposed a model of gene expression known as operand model. So today we'll study about operand model. So Jacob was genetist and Monod was biochemist, and they proposed a model in 1961 upon the operand model. An operon is a group of genes that are transcribed at the same time. Means operon is a coordinated group of genes such as structural gene, operator gene, promoter gene, regulator gene, which function together and regulate a metabolic pathway as a unit. For example, lacoperon, tripoperin, araoperin, and valoperin. So we'll study only lacoperon, which is in our syllabi. So first of all, what is the function of structural gene? Structural gene transcribes messenger RNA for polypeptide synthesis. Operator gene, it is a gene which receives the product of regulator gene. It allows the functioning of operon when it is not covered by the biochemical produced by the regulator gene. It function of promoter gene provides attachment site for RNA polymerase. You have studied about RNA polymerase, that it was used in the transcription process. The next is regulator gene. It synthesizes a biochemical or regulator protein which can act positively as activator and negatively as repressor. It controls the activity of operator genes. So repressor and inducer are or co-repressor are also found. Operator promotes and regulatory genes constitute the regulatory region. The lacoperon consists of three structural genes. First one is LAC-Z. The Z gene codes for beta-galactosidase, which is primarily responsible for the hydrolysis of disaccharide. Lactose into a monosaccharide units galactose and glucose. Next one is LAC-Y. The Y gene codes for permease, which increases permeability of the cell to beta-galactosides. And third one is LAC-A. The LAC-A gene codes for transacetylase, which can transfer acetyl group to beta-galactosidase. So Escherichia coli can use either glucose, which is monosaccharide, or lactose, which is disaccharide. But lactose is to be hydrolyzed first into galactose and glucose. So in this slide you can see promoter gene, regulator gene, operator gene, structural genes, etc. Structural genes I have told you in the previous slide, LAC-Z, LAC-Y and LAC-A, and functions of there I have told you earlier. Here again you can read the functions of regulator gene, structural genes. Structural gene, as I have already told you, LAC-Z codes for enzyme beta-galactosidase, LAC-Y codes for lactose permease, and LAC-A codes for enzyme thiogalactoside transacetylase. The regulator gene produces a repressor messenger RNA, and that is going to produce a lac repressor. That is, and this repressor will join with the operator gene, and the in the absence of lactose, this operon will be switched off. So repressor binds to the operator region and prevents RNA polymerase from transcribing the operon, and this is called switched off. The operon is switched off when repressor protein produced by regulatory gene binds to operator gene. RNA polymerase gets blocked, so there would be no transcription. So this is called switched off. So in this way, the Escherichia coli bacteria that cultured in the absence of lactose media would not be able to produce enzyme beta galactosidase, which is necessary for lactose metabolism. As we all know that regulator gene is going to produce repressor messenger RNA, and repressor messenger RNA produces repressor. Now if the inducer is present, lactose is present as an inducer, so repressor will combine with the inducer and it will inactivate the repressor. When it will inactivate the repressor, then RNA polymerase enzyme will activate the structural genes ZYA, and lac messenger RNA is formed, and then beta galactosidase, permease and transacetylase are formed. So if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of permease. A very low level of expression of lactoprone has to be present in the cell all the time, otherwise lactose cannot enter the cells. In the presence of an inducer such as lactose or allolactose, the repressor is inactivated by interaction with inducer. This allows RNA polymerase access to promoter and transcription proceeds. Now lactoprone when inducer when combines with repressor, means lactose when combined mixes with repressor, then the lactoprone is called switch on. Lactoprone is under control of positive regulation as well.